Okay, well, we'll start today for lecture 4, and this is on diagnostics and DNA sequence analysis. And so we're just going to start with something, an old technique, but one that illustrates a lot of background for a huge number of techniques which are used now, and that's ELISA, or enzyme-linked immunosorbent assay. And so just looking here on again, Google Images, we have a typical ELISA plate, and you can see what it is. It's a microtiter plate, and we're going to have different colors in each well, and the darker the color, the greater the presence of a particular antigen. So that's your end result. And so you can mark the plate any way you want with a Sharpie so that you can mark off different things. You usually want to have a control portion where you have nothing in there, no antigen at all. You'll have a control portions where you'll have no antigen and no substrate and so forth, and you have all those controls, and that'll be the background that you zero the machine up to. And of course you have a plate reader. You pop the thing in, and then the plate reader automatically gives you, you can tell it where the controls are, and it'll automatically, and then you give it a standard curve, so you usually run a standard curve on this as well, and then that'll automatically tell you how much protein that you've got in each well. It'll consult the standard curve, you input the values for the standard curve, for exactly how many milligrams per mil you have of your particular protein in your standard curve. And so maybe this over here might be the standard curve, and then you would just, the machine will automatically read that, and it'll calculate for you how much you have in your actual test, in your test runs. And you can run dilutions of the test runs if you want to, but normally you just put all your test runs in there, and it's done automatically. Okay, so that's great. So basically, the darker the color, the more antigen we have. We can go ahead and run a standard curve and figure out exactly how many milligrams per mil of protein, specific protein we have. The way this works is, if we look at this figure up here, what you've got is there are two different ways to do it. One way is, let's say that we're trying to find a particular antigen. Let's say it's a viral antigen from a sample. So you take that plate and you add to it this capture antibody right here. And that antibody is an antibody you probably made in your own lab. You know, maybe you've made it in mice or in rabbits or something like that. And you take that antibody, it's just a straight antibody, and you adsorb it to the plate. That is, you just go ahead and dispense into the plate the antibody. And you let it sit and incubate for a while, for a few hours, and then you just take the plate, flip it over, and bang it on some cloth so that you get rid of the liquid, but the antibody is adsorbed to the plastic. And then you wash it with buffer, and then you bang the plate again to get rid of the buffer, and then you're ready to go. The next step is you go ahead and you add the sample from a patient or from an organism. And you're trying to find, let's say in this case, a viral antigen. And you do the same thing. You let it sit, and you let it this time to bind specifically to that antibody. And then you wash it, and you get rid of the viral antigen. And then the next step is you go ahead and add a second antibody. And this will be the antibody that you had made in mouse or rabbits or whatever. But instead what we have is some sort of indicator molecule conjugated to your antibody. It could be radioactivity, or it could be a fluor, or it could be HRP or AP, so alkaline phosphatase or horseradish peroxidase, and from there we can use a colorimetric probe, so we can add a substrate that will turn a color in the presence of this enzyme. Or it could also be a chemiluminescence. It could be many different things. So in this case, let's say it's alkaline phosphatase, which is conjugated to your second antibody. Then all you have to do is add the substrate, the color of the substrate, and the alkaline phosphatase will turn that well yellow or purple, whatever your substrate happens to be. And then the degree of color of that well will determine how much antibody you have. So let's look at how that would look in the plate itself. And so we have, in the plate itself, it's going to look this way. You're going to go ahead and coat this, well actually, well let's go back, pardon me, because I have to give you the other way to do this before we can look at this. Okay, so going back here, we talked about being able to capture a viral antigen in patient serum. Now, this time, instead, this would be the top one. It's called a double antibody sandwich. The top one is if you wanted to see if a patient had a disease. That's what you're looking for. Does this patient have this active disease? Does this patient have this viral antigen in there? The bottom one is going to be, has this patient had the disease in the past? And in this case, you're going to see if the patient has antibodies against this disease. So for example, if I ever had yellow fever, then I would, and I've never had the vaccine for yellow fever, then I could go ahead, someone could take a sample for me from my blood, and then they could analyze it for the presence of antibody against yellow fever, because I still have those memory cells, right? So let's look for antibody in this case. So here what we do is we take a purified viral antigen, not from the patient, and this is ELISA, JP. So I take the purified viral antigen, not from the patient, but one that has been made in the laboratory. So it's a standard viral antigen. And then I coat the plate with that. Then I wash it out. Then I go ahead and add the sample. I'm not looking for viral antigen. I'm looking for the presence of antibody. Then I wash that out, and so that only this is sticking. All the dross is removed from the, all the contaminants from the virus, from the patient sample is removed. Just the antibody sticks. Then I add a special antibody, a secondary antibody, and this antibody is an anti-IgG antibody. It's an antibody against IgGs, against other immunoglobulin Gs. And it has an indicator, like alkaline phosphatase or horseradish peroxidase on there. And then you add the substrate, and the darker well, the more antibody must have been in that patient sample. Because the only way this antibody is going to stick is if another antibody from the patient has stuck to the antigen. That's the only way it's going to happen. Okay? So this one detects viral antigens, the top one. The bottom one detects presence of antibodies. So now we can go and look at this, because this one uses the second method. So here we're going to have a, we're going to coat this with viral antigen. And then you add the serum sample, and you're looking for presence of antibodies in that serum. And then we're going to go ahead and add the secondary antibody, which is an antibody against antibodies, and you notice it has these little curvy things on there, which is the horseradish peroxidase or the alkaline phosphatase conjugated to it. Then we add the substrate, and the substrate is converted by the enzyme into something colored. And so that's basically how it works. Well, any questions about ELISA? How do you use it? It takes a little while, but it works. I was investigating ELISA versus like PCR techniques for detecting pathogens, and I did a lot of work on that because I was thinking about creating some of these reagents by genetic engineering using plant viruses. So I looked into this. And it's very interesting. I thought that PCR would totally supplant the ELISA methods. And I read to the depth of even looking at discussion threads for diagnostics people. And it turns out for some diseases, ELISA actually works better than PCR. I thought PCR would be totally eliminate ELISA, but it's still being used. It's just better than PCR in some cases. In other cases, PCR is better. Okay. Now we'll go to monoclonal antibodies. How to produce monoclonal antibodies. Okay. Okay, so here we have, we're going to have two different cells that we're going to fuse together to create a cell we call a hybridoma. So a hybridoma is two cells fused together, and that's what we want. In the end, we want a hybridoma which produces antibodies. So what monoclonal antibodies are is they are antibodies produced by these mammalian cell lines, and the mammalian cell lines are true cell lines. That is, they are immortal. So once you create them, they will always be able to produce this one particular antibody. So polyclonal antibodies are when you inoculate a rabbit and with one particular antigen, but that on that antigen there are many epitopes, as you're all familiar with from immunology. So you get an antibody for every epitope. And so the polyclonal antibody will recognize for sure that protein, but it will have many antibodies, individual antibodies, each one recognizing a different epitope. And so it's just a crude serum that you isolate from the rabbit, and you use that as your polyclonal antibody. A monoclonal antibody, in contrast, is just one single antibody type which recognizes one single epitope on a protein. The nice thing about that is that you can do protein structure studies. You can say, well, we've got these 8 different monoclonal antibodies, we have these 8 different epitopes, and when we change this this sequence in the protein, this epitope disappears, and you can get into those sorts of things. Or we can say that this monoclonal antibody prohibits the protein, which might be on a virus, keeps the protein from binding to the cell. So therefore, that epitope that the monoclonal antibody binds to must be very important in viral attachment to its receptor. So you can do that kind of structural study with monoclonal antibodies, which is great. The second thing that's great about monoclonal antibodies is you only have to bother the mouse once, or the bunny. Because once you get basically the spleen from this thing, the spleen cells, that's the end. Because you can keep on growing these things over and over again, and you don't have to keep on injecting the bunny to get more polyclonal antibodies, because you have the cell line. So let's see, those are the advantages to monoclonal antibodies. Let's see how they work. So you do indeed have to go ahead and inject your mouse or your bunny with the protein. And then its spleen cells will produce plasma cells, which are going to be secreting the antibodies. Remember, so we have the B cells, and then these mature into the plasma cells that are actually secreting the antibody. So we have all sorts of different B cells in there, and we have some which are just reacting to some other antigen that's not in our, that we're interested in, some other protein. We have others that are responding to our protein, but we have a whole panoply of different ones which respond to different epitopes. So those are the spleen cells. Why don't we just grow the spleen cells out? What do you guys think? Why don't we just why don't we just go ahead and grow those spleen cells out? What's the problem with that? We could freeze them and then bring them back to life and use them anytime we want. We could grow them, for as long as we want. Why don't we use the spleen cells? What if I just took like your cheek cells and grew them? What would happen? Would I still have the culture a year from now if I were very faithful and I passaged them every 3 days? Why not? What's wrong with your cells? So Charles, I took your cheek cells. I very faithfully passaged them every 3 days, gave them new medium. Why would I not have anything after the end of a year?
They'll eventually die.
Well, yeah. Why would they die? Or why would they not passage?
They can't replicate after their telomeres are.
That's right. Their telomeres shorten, so they're mortal. They're not immortal. So that's the problem. So somehow we have to immortalize these guys, and the way you do it is fusing them with a cell which is immortal. And so they keep a group of, there's a small town in Iowa where they have a whole village of people who are immortal. And they supply the immortal cells for the rest of us. These people have been there for 400 years. No. No, I'm just kidding. No, you can get immortalized cells. And so, for example, myeloma cells. Myeloma, gosh, what is that? Oma, what would that be? Some sort of cancer, so it's immortalized, okay? So it's immortalized cell line. So we'll take those, and then we'll fuse them with the spleen cells, and we'll create hybridomas. Now, the next step is we have to somehow differentiate between the hybridomas and the spleen cells and the myeloma cells. So that's the trick. Once you fuse them, how do you separate them? Well, the plasma cell, the plasma cell from the spleen, all you have to do is replicate that enough times, and just keep on replicating, and eventually they all die out because they're not immortalized. Easy to get rid of. So the next question is, how do we get rid of the myeloma cells? Well, the myeloma cells are mutated. So they are mutated so that they do not produce HGPRT, which is a particular enzyme which is involved in the production of thymine, As well as HGPRT, thymidine kinase could be used as well. Thymidine kinase mutant or an HGPRT mutant would work fine. And what this means is that we're going to put these things in HAT medium. And HAT medium is going to, it stands for, well, let's go ahead and type it in here. It's hypoxanthine, aminopterin, and thymidine. And I'm going to get up here, and we'll just type in HAT medium. And this will be very funny when we put in HAT medium, which you get out of Google Images. You get a lot of hats. But let's go ahead and ignore the other hats and look at our HAT medium here. So in this case, instead of HGPRT mutant cells, we're using thymidine kinase mutant cells, but it's basically the same thing. So you use HAT medium in order to get rid of mortal cells and also get rid of your immortal cells and just keep the hybridoma. So that's what HAT medium is used for. It could be used in a lot of different ways. So for example, we have a mortal splenic B cell, and it dies. It eventually dies because it has a limited number of replication cycles. It does fine in HAT medium, but it is unable to go any further. So HAT medium has aminopterin. Aminopterin is going to block the de novo pathway for nucleotide synthesis. Aminopterin blocks the de novo pathway for producing nucleotides. Now, A's and G's are what? What are adenine and guanine? What do we call those? Purines. And then cytosine and thymine are pyrimidines. As you could guess, they're different structures. They have different biosynthetic pathways. So what we do is you have the aminopterin, which blocks the de novo pathway for producing any nucleotide because they share a common root. Then we have the salvage pathway, which would allow you to make A's and G's. And you can make it from hypoxanthine. Hypoxanthine is just a couple steps away from becoming an A or a G. So if you give these normal cells, these mortal splenic cells, HAT medium, they will not be able to make nucleotides from scratch, but from hypoxanthine, they can make A's and G's because they have all the normal enzymes they need. Thymidine is supplied, because you don't want to destroy the thymidine pathway. So you just give it the thymidine, and from there you can make cytosine very easily. But if you are TK minus, or if you're HGPRT minus, what happens then is that you cannot convert hypoxanthine into A's and G's. You can't convert it into adenine and guanine. So if you put something in hat medium, if it's wild type, it grows. If it's HGPRT minus, or if it's TK minus, it won't grow. It'll die. And that's how you look at hybridomas. Your spleen cells are mortal, so eventually they die. Your myeloma cells can grow forever, but not in hat medium. They die. It's poison to them because they are HGPRT minus or TK minus. But the hybridoma together, it's both immortal and it also has the proper wild type gene. It will live. Okay? So that's basically how to make a monoclonal antibody. And this, I think, is disturbing. It says a lot about our society. When we put in hat medium, which is two common English words, what do we get? We get Gilligan. And then even more disturbing is these squid hats. And what's really disturbing, and then a chihuahua hat, which is really sad. But then look at this. There's another squid hat. Another squid hat. Yet another. And then we have a dog wearing a Shriner's cap. Another squid hat. Another squid hat. Another squid hat. There's another one. It's the same guy. And he's dominating our culture. So if you want to know the real reason for global warming, I think we know what it is right here.
Squid hats.
So, yeah, it's squid hats. Exactly.
The same kind of squid hat.
Yeah, same. He's responsible for everything.
No, I understand.
So the whole culture is falling apart.
It's horrible.